Hey everyone, welcome back. So I wanted to talk to you about why DNA might need to be copied. In other words, what is the reason that DNA has to be able to be copied by your cells? And why is the copying so important that it gets done correctly? Well, sometimes you get injured and you need to replace those cells that you've lost. So in order to replace those cells, you need identical copies. Also, if you think about an animal like a puppy growing up to an adult dog, those cells just don't increase in size. You need more cells to make more muscle and more bone so that the animal can increase in size. Every time this happens, we're going to need to make copies of our DNA. Now, you don't need to know this process yet. You'll learn about it in high school, but there's a process called mitosis, and it basically explains how a cell is able to be copied. Inside the nucleus, if it's a eukaryotic cell, all of that DNA that we have will be copied. Every single nucleotide, every single base gets copied identically. And then that DNA gets moved around by the cell and eventually separated, and the cell splits so that we get two brand new identical cells. So it's important that DNA can be read and copied. So if we were to go back to our diagram of DNA, in order to copy it, the DNA basically opens up. And if you remember, each of these is called a nucleotide. We learned that thymine, the base thymine, is complementary to adenine. So if I'm going to make a copy of this side of the DNA, I'm going to need another nucleotide with adenine to come in. And over here, I'm going to need a guanine. Since this is guanine here, I'm going to need a cytosine to come in. And for this side, I'm going to need a thymine. And of course, I'm going to need the same nucleotides to come in on this side to make another strand that is identical. Let's take a look. Notice how the nucleotides are pairing up. New nucleotides are coming in from the cell. And by the time we're done here, we have two identical copies. So this is what's going on inside the cell before the cell can split. It basically ends up with double the amount of DNA so that one entire set of DNA could be sent to the new cell while the old cell keeps the original set. So if we look at it a little bit more as if we were taking notes here, and normally in class I would have you write this down, DNA will unwind itself from that twisted ladder, and there's a special enzyme that helps to do this. And it exposes the DNA. As you can see, new nucleotides are brought in, and they match up. So this strand is starting to look exactly like this strand over here. And it's enzymes that come through and read all of this and put these new nucleotides in place. Ultimately, I have two new strands of DNA that are forming. And eventually, those two strands of DNA have now been made from the original. And you can see why I color-coded them to help you understand that each new set of DNA has one new strand and one of the older strands. So these special enzymes or proteins help to unwind the DNA, hold the DNA open, bring in those new nucleotides, form the new strands of DNA, and then they spell check it. They make sure there's no errors. And finally, they wrap the DNA strand back up. Now we're going to learn later on about why it's important to spell check. If there's any errors, the DNA won't be able to have the correct code for making the right proteins. So I'm going to give you some problems today that are going to ask you to come up with a complementary side of DNA. And I'm going to show you how this works. So if one side of the DNA is this, then what would the other side be here? Well, if one side of the DNA is an adenine, what is its complementary pair? It's thymine.